Well, there's two major technologies. One is a methylation array, a microarray based technology that's looking at methylation changes and measuring that quantitatively, right, between two samples. The other way is through bisulfite converted DNA sequencing.
Hey, smart scientists! Welcome to another episode on the Smart Biotech Scientist podcast. Would you like to know more about the latest innovations in next generation sequencing? Well, you are in for a treat today because I'm having a conversation with Jonathon Hill, who is the Vice President of Science and Technology and the co-founder of Wasatch BioLabs and an Associate Professor of Cell Biology and Physiology at Brigham Young University. And we will be talking about innovating genetic sequencing. Are you juggling the complexities of CMC development while trying to enjoy the beauty of biotech? Have you ever wondered if there's a way to simplify bioprocessing? Welcome to the Smart Biotech Scientist podcast, where we're diving headfirst into the very challenges you face. We're breaking it down, demystifying the jargon, and giving you the keys to unlock your full potential. I'm your host, David Brohlman, and I get it. With 15 plus years in the biotech industry, I face the same challenges you do. There's a way to simplify and streamline so you can remove complexity, you can skip trials and errors, deliver without delay your groundbreaking therapy to clinics and market, and still enjoy every single step. Do you want to learn how industry experts and I did it? Grab a cup of coffee and your favorite notebook and pen. Now is the time to take your bioprocessing game to the next level. Let's smarten up biotech. Let's discuss the limitations of the current methods. We have fantastic technology at our disposal and much innovation is happening. So where do we currently face obstacles and how can nanopore technology be a solution?
Yeah, so right now, kind of the gold standard for methylation status identification, well, there's two major technologies. One is a methylation array, a microarray based technology that's looking at methylation changes and measuring that quantitatively, right, between two samples. The other way is through bisulfite converted DNA sequencing. Now, bisulfite is a chemical that will end up causing the conversion of Cs into Ts. But methylated Cs are protected from that, and so they do not get converted. And you can then sequence that and identify whether it changed or not and identify its methylation status with that. All of these techniques that I'm aware of that use bisulfite and then something like Illumina sequencing are based on PCR. Our biggest limitation right now, I would say, is that it creates two sources of noise within this data. The first one is how complete was your bisulfite conversion? Because if it missed a base and didn't convert it to a T, it's going to look like it was methylated when it wasn't, and you're going to get a false call there. So you have to be really careful about the treatment and how that works. Also, bisulfite is known to damage DNA and cause breaks in DNA. And so we get poorer quality sequencing and shorter sequencing when that happens. On the other side, when we do the PCR, by amplifying that DNA, we get kind of a bias and stochastic changes in the quantification of the DNA. Stochastic changes you can't do anything about. And the bias comes because if you have something that's highly unmethylated, it's going to become all AT rich. And those just don't amplify as well as something that has more balanced GC content. And so you get this bias towards unmethylated DNA. In that case, you underestimate your methylation state. So what we found is by using nanopore technology in our case, but there's others that do it, we can do native methylation sequencing, which means we're not doing PCR. This is native DNA, and we're not doing bisulfite conversion. And so we're looking at the methylation marks themselves. And they've gotten the AI models for base calling up to 99.6% accuracy on these methylation calls, which is plenty high for what we want to do. The other advantage it gives us over, say, an array-based method is we get an entire molecule of sequencing. In nanopore case, often many kilobases long. And we can look across that molecule and look at multiple cytosines and their methylation state across that molecule and make a call about the entire molecule. And we found that getting that kind of contextual information is really important to making an accurate call. Methylation changes at CPGs tend to be regional changes. And so the neighboring bases should all do the same thing. And so let's just give an example of why this is powerful. If I have something and I see a signal on an array that says 90% methylation, I don't know if that was 90% of molecules were fully methylated and 10% were unmethylated, or if each one was 90% methylated, right? I do not know the difference because I'm looking at things individually. But if I look at the whole molecule, I can tell the difference between that. I can tell you if 90% of molecules were fully methylated or if all of the molecules were 90% methylated. And that resolution ends up being really important in sorting out things because biological methylation states are noisy. They're not perfect. And so we can sort that through, and that greatly increases our sensitivity and our accuracy. And so for us, it's taken what was kind of a rough picture that we could get and important data for sure, but it really brought a whole new kind of level of resolution to what we can see in clarity in the data that we're getting, which of course, by increasing the quality of that data, we now increase the accuracy of the AI models or regression analyses that we do after that. They say junk in, junk out. Well, good data in, good data out kind of thing as well. And so we can create very accurate estimations of methylation state status within the sample that we're looking at.
Is that specific to the nanopore technology only, or do other sequencing technology allow the same level of detail?
So right now, you can kind of think of three major technologies. There's the short-read Illumina type technology, PacBio sequencing, and nanopore sequencing. Illumina still cannot do a true native methylation call. They're doing things to try to improve that and make it more direct, but they're still not there. But both PacBio and nanopore can do that kind of sequencing. We've tended towards nanopore. There's several reasons for that. One is the simple one that I had more experience with it because we were using it for some genome assembly type work. The capital equipment costs are far lower. I know our viewers can't see it, but I have a small nanopore sequencer behind me on my desk. They're very inexpensive. They're very small. They're portable. And so that was an advantage as well. And they tend to be more tolerant to different sizes of DNA. PacBio is a little more restricted in the ideal size that they want. But both technologies can do it, and both technologies do it very well.
And how does the sample prep differ between these? Is that comparable or is it very different?
Yeah, nanopore library preparation is very, very simple. It's a ligation of adapters onto the end, and there's a couple cleanup steps, and that's the entire process. In my lab, we have undergraduates prep these libraries regularly, and they do a good job. They generally succeed at it. And so very simple. PacBio is somewhat similar. They're ligating onto the end. They have this kind of hairpin adapter that they're ligating on the end, but it's a very similar library prep to do that. Neither of them are difficult. Neither of them take a long time. And both of them are automatable on a robot. And so you can go in there and just set your sample in. The programs are made, and they'll run and do everything for you.
Where are we with respect to the number of bases we can analyze with these technologies? What is the typical length or the maximum length of a DNA strain, for instance, we can analyze efficiently?
Yeah, so that's a great question. On nanopore, which I'm most familiar with, they've reported reads up to a couple of megabases in length. And so very, very long reads. We regularly see average lengths of 30 to 50 kb in our DNA samples. And so length is no longer an issue. In fact, fairly recently, they've published a true telomere to telomere human genome. And that was done in part with nanopore ultra long sequencing, which gets hundreds of kb average lengths on that. There is a trade-off. You get more throughput if you have shorter reads and more gigabases if you have longer reads kind of thing. And so we typically like to do, when we do whole genome sequencing, a 10 kb average. For us, that's the ideal kind of mix between length of read and gigabase output on the machine. However, one nice thing that we've loved about nanopore sequencing, it is read length agnostic. And so we work a lot in cell-free DNA. Cell-free DNA is highly fragmented because it's in the process of being degraded in the bloodstream. And so it has an average size of more like 200 to 300 bases. And so it does fine with those reads as well. So it can do both short, long. In fact, in a cell-free fraction, you get a mixture of read lengths anywhere from 150 bases up to several KB long, and we get all of those sequences together.
Now, being able to analyze is one thing, but you need also to make sense out of the data you're generating. Where are we there today? Because we have AI, we have machine learning, we have all these fancy tools, and it's just the beginning. Can you tell us about the role of bioinformatics in next generation sequencing?
Yeah, so this is where I think the challenge is right now. We've gotten to a point where generating data is easier than analyzing data. That used to be different in our field, where it would take months and months to generate just a little bit of data, and then you could analyze it in an Excel worksheet over the weekend. But now that's flipped, where we can generate gigabases of DNA in 24 hours, and it takes months and months to figure out what to do with it. And so that's something that we're actively working on. But here's the cool thing. You talked about AI models, machine learning, those kinds of things. What we've found is that you have to pay attention to the data that's coming in. And there's two levels of that. First of all, you want as accurate as you can get, right? The less noise the machine learning model has to work through, the better, the clearer picture it can create. But on top of that, if we look at the entire genome, for example, and we're looking for cell-free DNA derived from a certain population of neurons, less than 1% of that DNA is going to be informative. Most of the DNA is not going to be methylated, or will be methylated the same between tissues. We're looking for those unique sites, say a couple thousand of them in the entire genome where it differs. And so what we've done is we've developed a technology to target and select those particular regions. And it's only those regions that we sequence. And so we do a broad kind of sequencing. We figure out what regions are informative, and then we go in and target those regions and only sequence those regions. And we found the signal is so strong in those regions that we don't have to do a complicated AI model with tons of data in order to get it. We can do simple linear regression or categorization analyses, and there's no black boxes. There's no mystery to it. We can explain everything that's happening and show data for every call that's made. And yet we can get greater than 99% accuracy on identifying the tissue of origin of that molecule. In fact, on as little as 1,000 reads, we can estimate the fraction of the cell-free DNA from a particular tissue to about plus or minus 1%. And so we've greatly reduced the amount of data we have to generate. We've greatly increased the accuracy of it by looking at the important parts, and we've simplified the analysis by selecting these regions, which in turn makes it so that we can process a lot more samples together. We can multiplex samples on the same run and increase throughput, decrease costs for our customers, all of those kinds of things. So that's been a huge advantage to focus not just on the cool data analysis tricks that we have, but to really focus on the quality of the information going in to those models. Now, we have done AI and machine learning models on it as well, and they tend to work. We just found that our signal, when we can do whole molecule methylation analysis, is so strong that you just don't need to. It's night and day.
So how is your company pushing the boundaries of NGS and the epigenetics research?
Our big innovation right now is this targeting technology that I just alluded to. Our technology is the only one that I'm aware of where we can do this targeting without any PCR. And because we don't use PCR, we don't need bisulfite conversion. We can do native DNA sequencing and native DNA methylation calling on select regions in the genome. And so by doing this, we're greatly increasing the accuracy and lowering the cost of these epigenetic studies. And so how I see people applying this is, as we go forward, doing a broad kind of reduced representation methylation sequencing, something to identify these regions of interest. But then when the researcher says, now I want to scale to a bigger sample set and I want to look at 100 patients or 200 patients, we can select the regions that are going to be interesting to them and quickly identify the targeting sequences and those kinds of things and set up an assay where they can look at just the areas of interest, multiplex a bunch of patients on a single run at 10% of the cost of doing whole genomes on everybody, and greatly increase the ability for us to look at large patient samples with high coverage and good accuracy of the sample. And so we think that that is really going to open up and broaden our ability to do these epigenetic studies, especially large scale ones where cost becomes a factor.
Jonathan, where do you see the next generation sequencing in 5 years from now? Because there's so much innovation going on the technology side, on the artificial intelligence side. I'd be curious just to know what we will be able to do in 5 years.
So in my view, I think, well, the immediate focus right now is to improve the accuracy of the long-read sequencing. It's interesting, several people that I know tried long-read sequencing, nanopore, PacBio, in 2016, 2017 when they were coming out, and the accuracy was just so low. There was like a 10% error rate that it was unusable. And we've already gotten it to where it's a 99% accuracy rate. So there's been a tenfold improvement there. And I think we're going to see another tenfold improvement over the next couple of years. And of course, the better the accuracy, the better the results we're going to get. Another thing I see happening is we're going to see the application of NGS technologies more and more in the clinical space. Right now, I think we're at that transition point where it's moving from a cool research tool into something that has really arrived at a stage where it can be used in your clinical diagnostics. So we're going to see more and more genomic data in electronic health records. And we're going to see, I think, increasingly epigenetic data in electronic health record. And those are going to guide AI models and machine learning models that get developed to help guide doctors and patients in their treatment decisions. And so for me, we are at a transitional point. I often compare where we're at right now with NGS sequencing and biotechnology in general to the software world in the early 90s, mid 90s. I grew up in the Silicon Valley in California, so I got to see that transition from kind of this niche product to a mainstream product that everybody was using. And I think in the biotech space, we're seeing the same change to where instead of sequencing is something people know happens to they see it impacting their health decisions. They see it in their everyday life and everybody has access to it. And so that's going to be the biggest transition over the next few years.
Well, that's definitely exciting. I mean, we have an amazing future in front of us. Jonathan, what is the most important takeaway from our conversation today?
I just want the listeners to know that I think epigenetics and sequencing are things that we all need to be considering in our research. How can we use these tools to help improve our drug development or our diagnostic or co-diagnostics for our drug? How can we utilize this technology moving forward so we don't get left behind? The other thing to know is that my company, Wasatch BioLabs, we're here and ready to help support and team up with people as they're doing their research and finding ways to leverage our targeting technology and native methylation sequencing in order to advance your science. And we really take a hands-on approach where we partner with you. Each case is different. We help you design your targeting assays based on your data. We help run those assays and give you that data. And then you get to decide what to do with that and how it might impact what you're looking at.
Yeah, that's fantastic. Where can people connect with you, Jonathan?
So the best way is through our website, wasatchbiolabs.com. Wasatch is the name of the mountain range behind us. So we named it after the beautiful mountains that we have here in Utah. It's spelled WASATCH. And you go there and there's a form that you can fill out showing your interest. And we have people that will contact you and set up meetings. I am often directly in those meetings. We like to make sure that there is a PhD scientist sitting down with you, talking about the science, and really making sure that we get the assay right for you.
I will leave all the information in the show notes, so please do reach out to Jonathan. And Jonathan, thank you so much for sharing your insights, and thank you for being on the show today.
Thank you so much. It's been a wonderful conversation. I've really enjoyed it.
Hey, smart biotech scientists. What did you think of this podcast episode? Please tell us by leaving a review. And perhaps you are wondering how you can wire in novel technologies in your development workflow. If this is the case, I'd like to offer you a free clarity call to help you get started. Use the link down in the show note and I'll see you on the call. All right, smart scientists. That's all for today on the Smart Biotech Scientist podcast. Thank you for tuning in and joining us on your journey to bioprocess mastery. If you enjoyed this episode, please leave a review on Apple Podcasts or your favorite podcast platform. By doing so, we can empower more scientists like you. For additional bioprocessing tips, visit us at smartbiotechscientist.com. Stay tuned for more inspiring biotech insights in our next episode. Until then, let's continue to smarten up biotech.